Hi everyone. So let me explain about tonicity. Tonicity. No, it's not tonicity. So for tonicity, we can call it as concentration of solution. Solution. So what is solution? Let me give you an example. Sugar solution. Let's understand how do we make sugar solution. It's very simple, right? We take water plus sugar, we add sugar to water and we get sugar solution. Water here is solvent and sugar here is solute. So solvent plus solute is going to give you solution as a short form. No, it's not from math. For this, for us to understand tonicity, we also need to know what is osmosis. Let's understand what is osmosis. Hi. So osmosis is basically the phenomenon in which water molecules move from their higher concentration to their lower concentration across the semi-permeable membrane. Let's get back to the drawing. So osmosis, let me take this tonicity in the center and explain the rest of the parts. So there are three types of solution that we have depending upon the concentration. One is hypertonic solution. So this word tonic comes from the tonicity, and hyper means above or more. You might have heard people saying he is so hyperactive. Opposite to that, you have hypotonic. Hypotonic. Hypo means below or low. You'll understand it more. And there is one more type, that is isotonic. Isotonic, you must remember isosceles triangle where two sides are equal. Well, same is the case with isotonic. The two sides are equal. Of what? Yeah, of what? Let's understand. So for this to be understood by us, we need to know osmosis. So as I said, osmosis is the phenomenon in which water molecules move from its high concentration to its lower concentration. So in case of isotonic solution, you're going to have the semi-permeable membrane and they're going to be movement of water molecules. But in case of isosceles triangle, as we have both the sides equal, same is the case with isotonic solution, because across this semi-permeable membrane, if 25 water molecules go inside, or rather go outside the semi-permeable membrane, imagine this to be the cell, and 25 then are also going to come inside, right? And hence there is going to be equal number of water molecules on both the sides. Let's see what happens in case of hypertonic solution. In case of hypertonic solution, if I take a beaker and in the beaker I take, say, RBC, okay, and I am putting this RBC in seawater, concentrated salt solution. So in seawater, if I put RBC, what happens to the RBC is the water from the RBC will move out. Remember, water moves out of the cell because it moves from more concentration of water molecules to lower concentration of water molecules, right? Similarly, if in case of hypertonic solution, I put freshwater fish, right? Then similarly the water will move out of the freshwater fish and the fish will shrink. I said the things in this case shrink. Why? Because water moves out. This is called as exosmosis. Opposite to that is hypotonic solution. In case of hypotonic solution, if I take RBC and I put it in, say, distilled water or tap water, water moves inside the RBC. So what happens? Correct. It will swell. So things when put inside the hypotonic solution, they will swell. Water moves inside, and it is called as endosmosis. Alright. In case of isotonic solution, as you know, same number of water molecules on both sides, so there is no change in the cell structure. Now the thing is, in case of water molecules going inside in hypotonic solution, it is swelling. You know, hippopotamus, it swells, becomes like hippo, right? Since it's swelling, there will be a time when it will burst open, isn't it? That's in the case of animal cell. But in case of plant cell, remember, we have the outer rigid membrane, cell wall. And since there is cell wall, it will not allow it to burst open. Similarly, if I put cell of the plant into this, then let's see what happens. So as you remember, it should shrink, right? So as I was saying, it should shrink, but what happens is, in case of plant cell, we have the cell wall which maintains its shape, and the cell membrane will shrink with the contents, that is the cytoplasm, etc. But the cell wall maintains its shape. This is called as plasmolysis. Let's remember, in case of hypertonic solution, there is going to be more concentration of solutes and less concentration of solvent, and for us solvent here is water, and hypo is opposite of that. So less concentration of solutes, more concentration of solvent, and you know, in case of isotonic solution is going to be same on both sides. I hope this was helpful. Thank you.